My name is Jennifer Laborada. I'm a fourth year at UCLA.
Hello, I am Jasmine Santos and I am a third year at UCLA.
I'm Justine Ramos and I'm a fourth year at San Francisco State University.
My name is Miriam Velez and I'm a second year at UCLA.
My name is Julie Sanchez and I'm a second year at UCLA.
My name is Paul Ayub and I'm a fourth year at UC Berkeley.
We are Team HODOR, and today we'll be presenting on homology-directed repair. An outline of our presentation: first we'll be covering what is a double-stranded break and how the cell detects double-stranded breaks, and then we'll be discussing in detail the processes of homology-directed repair and non-homologous end joining, and how the cell cycle chooses which repair pathway to undergo, and methods to induce double-stranded breaks, and lastly, therapeutic applications. So what is a double-stranded break? So it's when both strands of the DNA duplex are broken. This can occur either through endogenous sources such as free radicals or exogenous such as ionizing radiation. And double-stranded breaks are detrimental because they can lead to cell death if they're not repaired, and if they're not correctly repaired, they can cause deletions and translocations in the DNA, which is commonly found in cancer cells. So the cell has adapted repair pathways to resolve DNA damage, two of which are non-homologous end joining, also known as NHEJ, and homology-directed repair, also known as HDR. So first, how does the cell detect double-stranded breaks? So here we have DNA wrapped around nucleosomes, and we have a double-stranded break. So after a double-stranded break, the mRNA complex is recruited to stabilize the free ends of the DNA, and the mRNA complex has three main players. There's RAD50, MRE11, and NBS1. And the most important players are MRE11 because of its endo and exonuclease activity, and NBS1, which recruits serine threonine protein kinase known as ATM. So ATM usually exists in an inactive homodimer, but once it interacts with NBS1, it is autophosphorylated and becomes active. Now it can phosphorylate serine residues on downstream targets such as H2AX. So as an overview, we have the DNA damage sensor, which is the mRNA complex that will recruit a protein kinase known as ATM, and that will phosphorylate H2AX, and this will create a binding site for a plethora of proteins, one of which is another protein kinase known as CHK2. And both H2AX and CHK2 will modulate cell cycle proteins such as P53, CDC25, and BRCA1. And this will not only sense the presence of a double-stranded break, but also signal to the cell that it's reached a checkpoint arrest and that this must be resolved before the cell can proliferate. And there's three main outcomes. There's apoptosis, also known as cell death, or DNA repair, or senescence, which means that the cell has lost the ability to divide and grow.
So now that we have discussed how double-stranded breaks are detected, we will then discuss how they are repaired. We will talk about homology-directed repair, or HDR, and we will be going through two models of resolving homology-directed repair called the holiday repair model and the double-strand break repair model. HDR is a repair mechanism used by cells in the event of a double-stranded break. It is a relatively accurate way of repairing a double-stranded break because it involves the use of a homologous template, either a sister chromatid or a homologous chromosome. So the main steps of homology-directed repair after a double-stranded break is to first create a single-strand DNA with a 3' overhang by exonuclease activity, followed by strand invasion or strand exchange, where the 3' overhang attacks a homologous DNA duplex, and then the HDR pathway will actually split between double-stranded break repair and synthesis-dependent strand annealing. We will only talk about double-stranded break repair today because synthesis-dependent strand annealing does not form any holiday junctions, and it occurs in meiotic cells to form non-crossover products. So the next step after strand invasion is to extend the region. And then after you extend the region, there are two ways of resolving the junction. So the two ways of resolving the junction that we will be discussing today are double-strand break repair model and the holiday repair model. So after the double-stranded break is caused by an exogenous or endogenous activity, the mRNA complex that was mentioned earlier will actually move through the DNA about 100 or 200 bases away from the break site, and a protein that is part of the mRNA complex called the MRE11 will perform an endonuclease activity and cut on the strand of DNA that has its 5' end exposed to the break site. Then it will perform a 3' to 5' exonuclease activity to chew back on the nucleotides and create the 3' overhangs. Now that the single-strand 3' overhangs have been made, proteins called the replication protein A, or RPA, will bind to it to stabilize the free ends. And after a series of protein coupling processes, the human tumor suppressor gene BRCA2 binds to a protein RAD51 and facilitates the exchange of RPA and RAD51. This leads to further stabilization of the 3' overhang and creates a RAD51 single-stranded DNA nucleofilament. In the second step of HDR, the stabilized single-strand DNA from one end of the damaged duplex will locate and pair with its complementary strand in the related undamaged duplex. So RAD51 will actually catalyze this homology search by catalyzing the opening of the homologous DNA duplex. So the formation of the heteroduplex, a region in which single strand from one DNA molecule is base paired with a single strand from a different DNA molecule, will form a displacement loop, or called a D loop. So once the D loop is formed, homologous recombination can then proceed to the formation of the holiday junction.
In order to resolve the D-loop formation from strand invasion, an intermediate must be formed known as the holiday junction intermediate. The junction is formed once a break is induced. The complementary strands from the homologous chromosomes cross and undergo hydrogen bonding. The strands are then ligated by the DNA ligase, and this crossover forms the holiday junction. Since the holiday junction are between identical or nearly identical sequences, they lead to symmetric arrangement of sequences around the central junction. This allows a branch migration process to occur where the strands move through the junction point. Branch migration allows for the exchange of DNA sequences between two homologous chromosomes without any net change in base pairing. As a result of this migration, a heteroduplex is formed from recombination, in which DNA strands from different sources are annealed. The holiday junction can be resolved through cleavage and ligation. In this image, the junction is being resolved by first rotating the bottom chromosome to form a cross-shaped structure. The structure can be cut horizontally or vertically by an enzyme called resolvase. The cleaved sections are ligated and result in different products according to the orientation of the cut. If the cleavage occurs in the horizontal direction, the duplexes are separated without exchange of DNA outside of the holiday junction. These products are called non-recombinant or patch products. If the cleavage occurs in the vertical direction, the DNA outside of the holiday junction is exchanged or crossed over between the two parental duplexes. These products are called recombinant or spliced products. Now that we have the main steps of homologous recombination, there are two models listed for the initiation of the repair. The first, or the DSB model, relies on the double-stranded breaks in the DNA, while holiday model is initiated by the pair of aligned nicks. DSB gets repaired according to the cell cycle phase it may be in. Non-homologous end joining, NHEJ, occurs throughout the cell cycle, which is depicted in the shaded region, while HDR only occurs in the S and G2 phase. As a side note, NHEJ plays a significant role in the development of the immune system, because it is important during the G1 phase, due to its critical role in recombination and immunoglobulin class switching. There are two protein complexes that influence the levels of NHEJ and HR. The 53BP1 and RIF1 protein complex induces NHEJ, while BRCA1 and CTIP promotes HDR. The expression of CDK1 in the S phase leads to phosphorylation of CTIP, which favors HDR. But without CDK1, CTIP isn't phosphorylated. Therefore, RIF1 protein is activated and NHEJ occurs.
NHEJ is the most common repair mechanism a cell undergoes, as it can be activated in any part of the cell cycle as mentioned by Justine. In contrast to HDR, NHEJ does not require a donor template or a sister chromatid to repair itself. Instead, the overhangs are simply cut off and the strands are ligated back together. As a side note, because of the loss of nucleotides, NHEJ is not the preferred repair mechanism for gene therapy. The first step in repairing the strand via NHEJ is the recruitment of the Ku complex. The Ku complex is a heterodimer composed of the proteins Ku70 and Ku80. The two proteins form a ring-like shape and attach to both sides of the double-stranded break. If needed, the Ku complex can translocate to the interior of the strands to allow other Ku complexes to bind if the overhangs were longer. Once the Ku complex bind, it recruits the DNA-dependent protein kinases to both sides of the double-stranded break, creating a DNA PK holoenzyme. Together, they occupy no more than 30 base pairs of the ends of the double-stranded break. Once binded together, the reactivity of the DNA-dependent protein kinase is activated. So in essence, it needs the Ku complex to be activated, but in extreme cases where Ku is not present, the DNA PK can become activated if there is sufficient ionic strength for it to bind to the double-stranded break.
Once the DNA protein kinase is activated, Artemis is able to bind to the DNA protein kinase holoenzyme. Artemis is a nuclease, a type of enzyme which cleaves chains of nucleotides. On its own, Artemis is not active. Binding enables Artemis to become active due to the DNA protein kinase phosphorylating Artemis. Phosphorylation is the process of adding a phosphate group to a molecule. Now active, Artemis comes down and displays endonuclease activity by cleaving back the overhangs of the double-strand break on both ends. This can be seen as the cut step in this cut and paste described mechanism. Here is the resulting DNA double-end strands. Nucleotide bases have been removed, the ends are clean, and the two strands are ready to be ligated back together. A complex is then recruited to the double-strand break site, which will ligate the two strands back together. This complex is known as the DNA ligase 4 complex, and it contains 3 different proteins. The first is DNA ligase 4. Ligases are enzymes that ligate DNA or other substances. The second is the DNA repair protein XRCC4, which stands for X-ray cross-complementing 4 protein. The third is known as XLF, which stands for XRCC4-like factor. Together, these proteins will work together to ligate the strands back together. This can be seen as a paste step in this copy and paste described mechanism. This forms a new strand of DNA. As we can see, non-homologous end joining is not perfect. First, nucleotide bases are removed. The two ends of the DNA are brought together and a new strand of DNA is formed. This can introduce frameshift mutations due to indels, which are insertions and deletions.
Now that we have discussed the mechanism behind double-stranded breaks, as well as the ways in which to resolve double-stranded breaks, we will now talk about methods behind inducing and utilizing double-stranded breaks to edit genomes. Genome editing is the act of modifying, inactivating, replacing, or removing a gene via the action of a site-specific nuclease. A nuclease is an engineered protein whose sole function is to cut the DNA at a location specified by the experimenter. Inherent mechanisms of the cells are induced after the cut that lead to 2 distinct types of repairs that we had discussed earlier: homologous recombination, or HR, and non-homologous end joining, or NHEJ. There have been 3 evolutions of gene editing in the last 3 decades. These include zinc finger nucleases in 1989, TALENs in 2009, and the most recent CRISPR-Cas9 in 2012. Because of its ease of use, CRISPR-Cas9 method is the current go-to methodology. A zinc finger is a small protein structural motif that is characterized by the coordination of 1 or more zinc ions in order to stabilize its protein fold. Using zinc fingers, we can create a zinc finger nuclease, or ZFN, which consists of 2 functional protein domains. The first, a DNA binding domain, which consists of eukaryotic transcription factors, and it also contains the zinc finger. The second, a DNA cleaving domain, which is comprised of the nuclease domain of FOLK1. The FOLK1 restriction enzyme is responsible for the catalytic cleavage of DNA. Zinc finger nuclease technology utilizes a FOLK1 nuclease as the DNA cleavage domain, and it binds DNA by engineered cis2 his2 zinc fingers. Each engineered zinc finger recognizes unique nucleotide triplets, and a combination of 3 or more of these zinc fingers will provide for high specificity in sequence recognition. Fusion of a FOLK1 monomer and pairing of 2 such fusion proteins will induce dimerization and therefore activation of FOLK1. The activated nuclease introduces a double-stranded break between two zinc finger binding sites, which prompts recombination and modification of the genome either through HDR or NHEJ. Tau, or transcription activator-like effectors, are proteins secreted by xanthomas bacteria that can bind promoter sequences in the host plant and activate the expression of plant genes that aid bacterial infection. Using TALs, we can create transcription activator-like nucleases, or TALENs, which are similar to ZFN technology. The main difference is in the DNA targeting protein, which is the TAL effector, or tail protein. TAL proteins are composed of 33 to 34 amino acid repeating motifs, with two variable positions that have a strong recognition for specific nucleotides. Like ZFNs, FOC1 monomers are fused to tails. Pairing of two such fusion proteins, creating TALENs, will induce dimerization and activation of FOC1, which thereby introduces a double-stranded break. Finally, the CRISPR-Cas9 system is a prokaryotic immune system that confers resistance to foreign genetic elements such as those present within plasmids and phages, and provides a form of acquired immunity. CRISPR spacers recognize and cut these exogenous genetic elements in a manner analogous to RNA interference in eukaryotic organisms. There are two classes and multiple types for each class. For convenience sake, we will discuss type 2 of CRISPR, which is the most widely used. This utilizes the Cas9 protein within the system. In the original form, this system uses three components: two short RNAs, CrRNA and tracer RNA, and a Cas9 RNA-dependent nuclease. The CrRNA contains a 20 nucleotide target sequence, or the guide sequence, and it hybridizes to complementary sequences on the genome. The tracer RNA has sequence complementarity to the palindromic repeat within the CRISPR sequence. A complex structure formed between CrRNA and tracer RNA is then recognized by a Cas protein, which is brought to the target site and exerts its nuclease activity. For convenience sake, CrRNA and tracer RNA can be fused to make a single guide RNA, which maintains the original activity of CrRNA and tracer RNA. The genomic target can be any of 20 nucleotide DNA sequence, providing it meets two conditions. The sequence is unique compared to the rest of the genome, and the target is present immediately upstream of a protospacer adjacent motif, or PAM sequence. The PAM sequence is absolutely necessary for target binding, and the exact sequence is dependent upon the species of Cas9. This sequence from 5' to 3' is NGG. Once expressed, the Cas9 protein and the guide RNA form a riboprotein complex. This is done through interactions between the guide RNA scaffold domain and the surface-exposed positively charged grooves on the Cas9. The Cas9 then undergoes a conformational change upon the guide RNA binding that shifts the molecule from an inactive non-DNA binding conformation into an active DNA binding conformation. It's also important to note that the spacer sequence of the guide RNA remains free to interact with the target DNA. Once the Cas9 guide RNA complex binds a DNA target, a seed sequence at the 3' end of the guide RNA targeting sequence will begin to anneal to the target DNA. If the seed and the target DNA sequences match, then the guide RNA will continue to anneal to the target in a 3' to 5' direction. Cas9 will only cleave the target if sufficient homology exists between the guide RNA spacer and target sequences. Since all three technologies allow for genome editing, what's the difference? Both ZFNs and TALENs are not limited to mutagenesis in mouse embryonic stem cells. However, the interactions within TALENs are less complex than those within ZFNs, making designing TALENs as well as prices for TALENs much cheaper than those of ZFN. The CRISPR-Cas system offers also several advantages over ZFNs and TALENs. The first is target design simplicity. Guide RNAs can be designed readily and cheaply to target nearly any sequence in the genome specifically. The second is efficiency. Modifications can be introduced by directly injecting RNA encoding Cas proteins and the guide RNAs into developing mouse embryos, thereby increasing efficiency. And the third is multiplex mutations, where mutations can be introduced in multiple genes at the same time by injecting them with multiple guide RNAs. All three technologies offer researchers alternative methods to develop mutant mice and human disease models faster than any traditional gene targeting method, but these are not without their limitations and complications. They include off-site effects where mutations can be introduced at nonspecific loci with similar but not identical homology, as well as multiple alleles, where the healing of a nucleus cleavage site by NHEJ can produce mice with different mutations from the same targeting constructs. This can also produce mice with multiple variants that can create a phenotyping bottleneck.
Now that we've talked about homology-directed repair and methods to introduce double-stranded breaks, I will now like to talk about the application of this technology to edit genomes. Targets for genome editing are broad, ranging from the eyes to the liver, with our lab focusing specifically on monogenetic disorders of the bone marrow. A recent review from Molecular Therapy outlines all the active areas of preclinical research covering gene editing. Areas include liver-targeted gene editing, neuromuscular disorders, skin disorders, viral infections, T-cell immunotherapy, and hematologic disorders such as SCID and sickle cell disease, which are areas our lab actively focuses on. This paper from our lab is an example of a preclinical study demonstrating that hematopoietic stem cells from patients with sickle cell disease can be functionally corrected through gene editing. In this paper, ZFNs and a homologous donor template were co-delivered to genetically correct the sickle cell disease mutation at the beta globin locus. And here in this figure, you can see that when bone marrow cells derived from a sickle cell patient are treated with zinc finger nucleases and integrase defective lentiviral vector, their erythroid differentiated progeny produce less sickle hemoglobin and more adult hemoglobin, demonstrating the potential of gene editing technology to cure the most devastating of hemoglobinopathies.